Hi, I'm Sandra Porter, and today I'm going to talk about drug discovery using Protein Blast and IC3D. At the end of our last video, we were talking about our BLAST results from searching the structure database at the NCBI using Protein Blast. And in those results, I found a candidate that I want to investigate from the infectious bronchitis virus main protease. Okay, and this candidate is bound to an inhibitor, and the structure is called 2Q6F that I'm going to take a look at. And I had copied the alignment and pasted it to a Google Doc. You could also use a Word doc to work with later on. All right, what I'm going to do now is I'm going to click the link to take a look at my record, my structure record for this protease, and I can see that this protease entered the database December 1st, 2020, 309 amino acids long. I can see that this is related to coronavirus, not exactly the same, but it's part of the same lineage. I can see an article, a published publication about it, and I could look at this link if I wanted to learn more about it. I can learn more by looking here to see different annotations about the structure, learn more about the structure that way. And I can also click the link to see the two structures that were referenced in this publication. And I could see here I'm looking at 2Q6F, and I could look at that, to click the link, read the abstract, to learn more about the experiments that were done to discover this structure. And I can scroll down and see that there's two different chains of protein in this structure and two shorter peptides that are 6 amino acids long each. They're modified peptides, as you can see by the name. I could look at the sequences, and I can click full-featured 3D viewer to look at this in IC and 3D. Before I do that, I can also see how they're interacting. I can see there's one bound to chain B and one bound to chain A. Okay, so now I'm going to switch over to a different browser because I like to use IC and 3D with Firefox. And we'll take a closer look at the structure and figure out what it's doing. Okay, so I'm going to open the analysis menu and click sequences and annotations so that I can see the different chain names and see how these are organized. I find sometimes that there is information about the structure that is present in the first chain of the protein, but it's not always present in the second. So if I have more than one identical protein chain, I almost always pick protein A or chain A to work with. Okay, I can see, so I'm going to pick chain A, and I want to take a look at this inhibitor that's binding. So what I'm going to do is I'm looking at the brown one, because that's one that's binding chain A. So I'm going to click the name of the protein here, and you can see it highlighted it. And I'm going to save that selection, and I'll call it inhibitor so I can find it later on easily. Okay, I'm also going to identify the binding site for that inhibitor. So I open the select menu, I choose by distance. I'll choose inhibitor here in the first set, and we're going to look for anything that's within 4 angstroms of my inhibitor, which is fine. And I can pick chain A here and say display. Okay, so you can see that I have selected amino acids that are close to this short brown peptide here that I was looking at earlier. And again, I'm going to save the selection and give it a name that I'll remember. So I'm going to choose Save Selection. And I'm going to call this binding site. And you can see it's appearing here in my my sets. Okay, so I would like to know, first of all, whether or not the amino acids in the binding site are present in both this infectious bronchitis virus protease and the protease from SARS-CoV-2. So what I'm going to do is I'm going to move the sequence over a little bit and expand the window and click details so I can see the sequence. And now I can see which amino acids are highlighted and present in the binding site. So first I see there's a histidine at position 41, and it's preceded by a CPR. So I'm going to go to my alignment that I saved earlier and highlight that. So I can see CPR, and I can see the histidine, and I'll highlight that histidine. Oops. Actually, that wasn't. There we go. And it's great that histidine is present in both my query and the subject sequence. Now let's just stop for a minute and think about what these are. My query sequence came from SARS-CoV-2, and it was from the protein 7VH8, and I think chain A. I think I can do that. Here we go. My I think I can have a space there. My subject sequence that I'm just looking at now is from 2Q6F. Also chain A, because it's good to be able to keep track of which sequence is which. The query is always the one that I used to search the database, and the subject sequence is always the one that came from the database, something that I found. Okay, and this is kind of nice because both of these sequences have histidine at position 41, and you may remember from our first video that histidine was one of the catalytic amino acids that's involved in the protease activity. So that's important that it's present in both of these. Okay, let's go back and look at the others. All right, there's a lysine at position, let's see, position 45, and it's right after a VLG. So I'm going to highlight that. Okay. There's the VLG, and there's the lysine. And you can see that one is present in the infectious bronchitis virus, but it is not present in the SARS-CoV-2 protease. And let's scroll here and see some of the others. Okay, I've got an HLEL here that begins at it's histidine 162. So let's take a look here. We're looking for HLEL around 162. And I can see it right there. Now I can see that histidine 162 is present in both of my proteases. As are the glutamic acid and the leucine. I'm going to zoom in a little bit here so you can see this a little bit better. Now, right here, there is a plus in the alignment, in the center of the alignment, and that plus is telling me that methionine is conserved. So it's not identical to the leucine, but it is similar. So we'll give that a highlight, but we'll give it a somewhat different color. Okay. Maybe we'll give it a little bit more of a color that stands out a little bit more. There we go. Okay, so what else have we got? The other sequences that we have that in the binding site, there is an aspartic acid at 185. Looks like it's DEEV. Okay, we're looking at DEEV, which I've got right here. I'm just going to go back and look at this for a minute. And then there's an A, an alanine, and then there's a Q at position 190. So let's get that too. Okay, and I can see the D, aspartic acid, is present in both of my sequences. S, there's a Q here, and it's saying that this is conserved, so I'll give it a slightly different color. And the Q is present, the glutamine. Okay, so I can look at this and I can figure out how many of the binding site amino acids are present in both of the structures. If I count them, there's a 1, 2, 3, 4, 5, 6. Okay, so 6 are identical. I can see that if I count the conserved ones, I've got 8 are identical or conserved. And how many do I have total? I had 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 total. So 8 are identical or conserved out of 11. Out of 11 total, which is great. Now what I'd like to know is which of these amino acids are forming bonds to the inhibitor, and are the amino acids that form bonds present in both my structures? So we're going to take a look. Okay, the first thing I'm going to do, I'm going to make this a little bit smaller. There we go. Make my structure window a little bit larger. So I can see a little bit better. And I'm going to annotate the binding site so that I can look at interactions between the binding site amino acids and the inhibitor. So what I'll do first is I'm going to take, I'm going to open up my defined sets here. First, we'll look for covalent bonds. So I can select the inhibitor, and I can also now hold down the shift key and select the binding site. So both of these are selected, and I'm going to change the drawing style and make these ball and stick. Because if I do that and there's a covalent bond between them, that bond would appear and I'd be able to see it. This is also kind of nice because it shows me, where the binding site is in the protein, but it's on the outside. Now I'm going to hide everything else so that I can focus on the interaction here between the inhibitor and the binding site. So no covalent bonds, but what other kinds of bonds might there be? So what I'm going to do now is I'm going to open up the analysis menu, and I'm going to choose interactions. And the bonds I'm going to focus on today are, I'm going to focus on hydrogen bonds and salt bridges. So I'm unchecking everything else. And what I'm going to do is select the binding site, and the inhibitor. And it doesn't really matter which order we do this in. And then I'll say 3D display interactions. Okay. And what this is showing me is that there are 4 hydrogen bonds between these molecules. And I can tell they're hydrogen bonds because they're kind of a bright green color, and there's a bright green color here in the key. Okay, so now what I'd like to do is to figure out what amino acids are binding here. Okay, so what I can do, and kind of like maybe what parts of them are interacting. I can hold my mouse over here and I can see this is a glutamic acid at 187. And it's binding here to this section of the inhibitor. I can also take this and I can make this, color this by atom. Give me a little bit better picture of how the hydrogen bond is forming. So you can see there's the carboxyl oxygen from the side chain of glutamic acid, and here's a nitrogen in the backbone of the inhibitor. Okay, so glutamic acid 187. Let's see if that's what we can see about that. Okay, so glutamic acid 187, 1, 2, oh, actually, should see that in my sequences and annotations. Oh yeah, there it is. So it's the E in the DEEV. All right. Okay, so that is this one right here. And it's not exactly the same, but remember this one is also conserved. So let's just mark this so that I can remember that that was forming a hydrogen bond. And let's see what else we've got. That was glutamic acid 187. We have also forming a hydrogen bond. We have the backbone, backbone oxygen from valine 188. So that was the V. And that one is part of the binding site, but not found in my SARS-CoV-2 protease. Let's see what else we have. Here we have glutamic acid 164, and this is also glutamic acid 164. Interestingly though, you can see that here's the side chain from the glutamic acid with the carboxyl group, and it's the oxygen and nitrogen that are part of the backbone that are forming the hydrogen bonds to my inhibitor. So glutamic acid 164, which seems to be really important. And that is, oops, let's take a look here. 164, right, it's right after the HL. It's the E right here. Okay, so it's this one. So let me highlight that guy. Okay, so I can see that so E164 is forming, has a hydrogen bond, or is present in both proteins, forms a hydrogen bond. I see that glutamic acid 188 is only present in one, but it's possible that the but the glutamine might be able to form a hydrogen bond. And that actually makes a lot of sense if you think about it, because if we look at this, the side chain that is interacting with the inhibitor has a oxygen here, right? 2 oxygens at the end of the side chain. Glutamine has an oxygen and a nitrogen. So it makes sense that glutamine could be forming a hydrogen bond at that position. All right, so what do I conclude? I conclude that it is likely that the inhibitor could also bind to the SARS-CoV-2 protease. One of the important amino acids for forming a hydrogen bond is present. Actually, possibly there's one that's conserved that could be forming a hydrogen bond that's present. And this one is actually forming 2 of the hydrogen bonds. So 3 out of the 4 hydrogen bonds could be formed. And I also know that 8 out of the 11 amino acids in the binding site are present. So I'm going to go with, this is pretty likely that it's going to be able to bind the inhibitor. And that's it. Thanks.